says E. coli requires three genes: Gal-K, which is a kinase, Gal-T, which is a trans-acetylase, and Gal-E, which is an epimerase to utilize galactose. Like these, you can understand by the statement that it is a these are all galactose operon. Genes are from the galactose operon, which has three genes, and all these genes, the three genes are required to utilize galactose or metabolize galactose. If there is a mutation in any one of these genes, then the mutant cannot utilize galactose. So mutant will not be able to use galactose if any of these three genes are mutated. Which one of the following combinations of merodiploids will support the growth of mutants on galactose? So they are asking which of the following combinations of merodiploids will support the growth of mutants on galactose. Okay, galactose is the only sugar source here. And so merodiploids concept I had already explained in the previous lectures. I'll just go through a little revision. So in bacteria, there happens a kind of transfer of genes, or this bacterial reproduction takes place when there is a bacteria which has the F plasmid. So it is whatever bacteria has the F plasmid, they are called the F plus cells, and these F plus cells can form the conjugation tube. They uh, they will what they can do is like they can form the conjugation tube, and they can transfer the F plasmid to an F minus cell. But sometimes what happens that the F factor can integrate into the bacterial chromosome. Okay, F factor gets integrated into the bacterial chromosome, and the cell becomes an HFR cell. HFR is a high frequency recombination cell. So when there now like it was integrated into the bacterial chromosome, it can also disintegrate or excise out from the bacterial chromosome when it has to initiate the reproduction. So sometimes it happens, sometimes there is a normal excision, no other genes, no other bacterial chromosome or bacterial genes will be a part of the F factor. But sometimes uh, excision of the F factor, what happens is that there is additional uh, genes from the bacterial chromosome come excise up. So here you can see along with the F factor there are other genes like A and E. They are also coming out in the with the F factor. So now this becomes an F prime cell. So now this when this cell starts starts to form a conjugation tube, sometimes it happens that along with the F factor, the other A and E chromosomes, they are also like A and E genes, they are also getting transferred like here. So in this bacteria, F prime cell, along with the F factor, it will have two copies of two copies of the A gene, which are in two different forms. Like you can see capital A and small a, and E is also of two forms, capital E and small a. So because this bacteria has two copies of the same the two forms of the same genes. Okay, they are partially diploid for these two genes only, and therefore they are called merozygotes. So here also the same thing has happened in the question. They are telling the merodiploids are there. So merodiploids, only those merodiploids will be viable in galactose media in which all these three genes are present. These three genes have to be present. So let's see. Here it is saying in option number one, so gal K is there, plus signs indicates that the gene is a wild type gene, and minus sign will indicate that it is a mutant or mutation is there present. So gal K is there, gal T is there, and gal E is not there. So And in the, if since it is a heterozygote, so it will have another copy of the gene. So here also gal K is not there, gal T is there, okay, and gal E is not there. So in both, in this entire combination, you find only gal K and gal T are present, okay, and gal E is not present. So since gal E is not present in this entire combination, it will not be able to metabolize galactose. So let's see the other option. Q, Q gal K is not there, gal T is there, gal E is not there. Okay, gal T is there, and in the other combination, gal K is there, T is not there, and gal E is there. Since in this entire combination, all these three genes are present, so this will be able to metabolize galactose. Let's see the next option. Gal K is there, T and E is not there. Here K and T are not there, E is there. So K positive, E positive, but T is not positive, so it will not be able to metabolize galactose. Last option, K is there, T is there, E is not there. K is there, T is not there, but E is there. So all the three genes are positive, at least in one copy. So this will also be able to metabolize galactose. So Q and S will be the combination. So here Q and S is D. So this combination of neurocamploids will be able to support, will be able to grow utilizing galactose as the carbon source.